Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 11th December 2020. And as all of us know, we have been revising the entire syllabus after having completed the videos, and then we are now revising chapter number 14, Respiration in Plants. Our memory helps us in tracing out what we have been revising. First, step number 1, glycolysis. And we understood that through glycolysis, there is release of energy of 8 ATP molecules. And then the glycolysis is followed by step number 2, that is Krebs cycle. And in the Krebs cycle, also a lot of energy molecules are generated. To be precise, 24 ATPs are released. So glycolysis 8 and Krebs cycle 24, so 32. And then third step, electron transport chain. And that also a lot of ATPs are released. And overall, if you see the balance sheet of ATP during complete oxidation of one molecule of glucose, in glycolysis as such happens in cytoplasm of the cell. So 2 ATPs are released and formation of NADH, 2 NADH. So each NADH molecule is equivalent to 3 ATPs. So 2 into 3, 6. So 6 plus 2, 8. And then formation of acetyl-CoA in matrix of mitochondria. Again, there is 2 NADH are formed, that is equivalent to 6. And then in mitochondria, the matrix 2 ATPs and 2 FADH2. So 1 FADH molecule is equivalent to 2 ATPs. So 2 FADH2, 2 into 2, 4. And then in Krebs cycle, of course, 6 NADH molecules and 6 into 3, 18. So totally through ATPs, glycolysis 2 ATP and mitochondrial complex 2, 2 plus 2, 4 ATPs directly are formed. And through NADH and FADH2, it's almost say 6 plus 6 plus 4 plus 18, 32 or 34 ATPs are formed. So total net gain of ATP molecules because of breakdown of just one glucose molecule is 38 or 36 depending upon the type of aerobic respiration. So in ETC, electron transport chain, that we were reflecting upon yesterday, we said there are 4 complexes. One is the NADH coenzyme reductase complex, or it is also called NADH dehydrogenase complex, that is complex number 1. Complex number 2 is succinate dehydrogenase complex, and the third one is cytochrome BC1 complex, and the fourth one is cytochrome C oxidase complex. And there is a fifth complex, it is called Atp Synthase complex, which involves the ATP synthesis. This is how the ATP synthesis will take place. Oxidative phosphorylation is the synthesis of energy-rich ATP molecules with the help of energy liberated during oxidation of reduced coenzymes. What are the reduced coenzymes? There are 2, NADH and FADH, produced during respiration. The enzyme required for this synthesis is called Atp Synthase or mitochondrial ATPase. It consists of 3 parts. F1 particle, as you are seeing, this is the F1 particle, which is the headpiece of the projection observed on the matrix side of the mitochondrial crest. F1 contains 5 associated subunits, and F0 complex, this one, is localized in the membrane bilayer and containing the proton translocating mechanism. It is composed of 3 to 4 distinct polypeptides and 1 protolipid, a protein stack that connects the 2. This is the protein stack that connects F0 and F1. The protons, what are protons? H+. These are the protons. originating from electron transfer are translocated by the respiratory chain across the membrane from mitochondrial matrix side to the cytosol side or outer side, from M side to C side. Okay, and this helps in pushing three pairs of protons to the outer chamber, while two pairs of protons are sent outwardly during electron flow from FADH2 to the ETC, that is, as the latter donates its electrons further down in the electron transport chain. According to chem isomatic hypothesis, this translocation creates a pH difference. So the movements in the protons creates a difference, and that pH difference and a membrane potential both constitute proton motive force that tends the movement of H plus from C side back to the M side of the membrane. Since the inner mitochondrial membrane is highly impermeable to H plus ions, these can only reach the M side through the proton channel of the ATPase. When H plus moves from C side to M side, the F1 ATPase operating in reverse catalyzes ATP synthesis. This is how ADP plus inorganic phosphate in the mitochondrial matrix ATP is synthesized. The flow of protons through F0 channel induces F1 particle to function as ATP synthase. The energy of the proton gradient is used in attaching a phosphate radical to ADP by high energy bond. This produces the combination of ADP plus inorganic phosphate, ATP. The transfer of energy from electron flow to electrochemical proton gradient and hence to the phosphate bonds of ATP is called oxidative phosphorylation. So what is the difference between oxidative phosphorylation and photophosphorylation? Oxidative phosphorylation, it occurs in respiration process as we discussed, and photophosphorylation, it occurs in photosynthesis. Oxidative energy of oxidation reduction is used for production of proton gradient required for phosphorylation, that is ADP plus inorganic phosphate. Light energy is utilized for the production of proton gradient for photophosphorylation. So here it is the light, use of light, here it is the proton gradient that is used. So the energy released during ETC is used to make ATP with the help of ATP synthase. This is the enzyme required, which consists of two major parts, F0 and F1. F1 is the peripheral membrane protein complex having site for synthesis of ATP from ADP and inorganic phosphate. F0 is the integral membrane protein that form channel for the protons. For each ATP produced, 2H plus passes through F0, as you are seeing, as it passes through F0 from the intermembrane space to matrix down the electrochemical proton gradient. So you are seeing here the inner mitochondrial membrane. So what is the difference between fermentation and aerobic respiration? Fermentation, it accounts for incomplete oxidation of glucose as we discussed, and aerobic, it complete combustion, complete oxidation of glucose. In fermentation, there is net gain of only 2 molecules of ATP, just 2 ATPs. In aerobic, just now we discussed, there is something like 36 to 38 molecules depending upon the type of aerobic respiration. And NADH is oxidized to NAD plus very slowly. NADH is oxidized to NAD plus very fast, as we see. So, amphibolic pathway. Glucose is the favored substrate for respiration. All carbohydrates are usually converted into glucose before they are used for respiration. Fats needs to be broken down into glycerol and fatty acid, which is further converted into acetyl-CoA, and enter the respiratory pathway. So carbohydrates are converted to glucose, fats are also converted. Now proteins. Proteins are broken into amino acids and further enter into Krebs cycle. That means whatever food that we eat having carbohydrates, proteins, fats will all be entering into Krebs cycle. Thus we are getting energy. So breaking down process within living organisms is called catabolism, and synthesis process making is anabolism. Since respiration involves both catabolism and anabolism, it is called amphibolic pathway. Hence, through respiration, we are getting a lot of energy. Thus, it is always beneficial for us to have lots of glucose. Hence, our effort should be to have